Thursday evening to come in here at the Laureate Psychiatric Clinic and Hospital. We're starting, this is the inaugural first lecture on the Healthy Brains for a Healthy Oklahoma series. And you probably saw the flyers, and we'll be doing this series once a month, every fourth Thursday. And we'll cover a number of different topics. And what we really like to do is engage you and the community at large in kind of thinking about the brain, what it means for people growing up, for little people, for older people, and what we've learned so far and how we can use that knowledge to look at people a little differently. This is part of an effort that the Laureate Institute for Brain Research, or LIBR, is doing to give back to the community, to basically show what we have done and what we're going to do in brain research in Tulsa. The Laureate Institute is really an absolutely unique institution in the country. It brings together some of the best neuroscientists in the field, trying to discover new tools and techniques to help people with mental illness. And as part of that, we will be one side of a large study that will start this year to study between 10 and 12,000 kids, ages 9 to 10. And the goal of this study is really to try to understand the developmental trajectory of the brain during adolescence and what happens in the brain, what is the implication of that for various types of functions of the brain in terms of learning, in terms of how we relate to each other, but also in terms of the risks that a young brain faces in terms of drugs and alcohol, in terms of depression, in terms of anxiety. So this study will be something that I think will really put Tulsa on the map in a larger scale in terms of brain research. And we're very excited about this, and you will hear more about it as the study progresses. But today I'm just going to give you an overview of how the brain grows up. Let me say from the get-go what the presentation will be and what it will not be. So what I really wanted to do is give a presentation for people who have basically no background in neuroscience. So you can come in here and never heard anything about the brain at all, and hopefully you'll get some insights as to what we know about the brain. So I will start very, very simply in terms of what are the components of the brain, and then lead you through what happens during development. I will focus mostly on healthy development, so we will not be discussing any pathologies or abnormalities that happen during development. We're hoping to do that sometimes in the future. And of course, as you can imagine, there's so much to cover that I probably will leave some of the things out, maybe some of the things that you're interested in. What I'm really hoping for is that if you find this interesting, if you want to come back, make suggestions to us of what you would like to hear about, so that we can really bring to you the knowledge that you would want to know more about. So, this is really a back and forth. And then finally, at the end, we'll have some times for questions and answers. I'd be happy to answer questions. If there are questions and if something is not clear, even during the presentation, just let me know. I'm perfectly happy to take questions anytime and try to clarify some of the things that I'll be talking about. As far as I can tell, the lecture will be about 45 minutes to an hour, roughly. So we have plenty of time if we want to talk about things afterwards, or we can just go home early. Again, what I will do is really try to avoid any time of extensive jargon and terminology. I use some words, but hopefully I will explain all of those as we go along. Okay, so without further ado, let me just get started. So let me give you first an outline of what I'll be talking about. And then I will go through each of these points. So we'll start very simply with what I call the brain and its parts. And what I'll do there is basically tell you what we know about sort of the general brain, what are its components, and that will be important because then that will help you understand what happens during development. And then we'll talk about how the brain changes from birth to adulthood. And that will be followed by something that's really interesting, I find extremely interesting, which is a term called critical periods. Critical periods, basically what that means, we understand now that the brain has windows that open and close. And these windows that open and close through a certain machinery that is in our brain. And what that means is that during certain periods of time, the brain is better able to learn and acquire new skills than during other times. So I will talk a little bit about what opens the window and what closes the window. I think that there will be some very practical implications of this. Then I will talk about stress and development. So that's the other side, which is what impinges on the brain that may potentially make it more difficult for the brain to develop and grow up. And then finally, I will be talking about hot and cold thinking. And that is particularly relevant to adolescence. And I'll make that very clear when we get to that topic. Okay, so the basics about the brain. So first, as you may know, the brain is about 2% of your body weight, but it actually consumes about 20% of the energy of your body, and therefore consumes about 20% of the oxygen that comes through your blood. So it's a very high energy organ. We know that the brain has about 80 to 90 billion neurons. Neurons are the nerve cells, and I'll talk more about them. So that's a tremendous amount of nerve cells. And we also know that the brain is actually mostly water and fat. The brain is the fattiest organ in the body, and it's about 2/3 dry weight is fat, and that has and there's a certain function that goes along with it, and I'll talk about that as well. The impulses, the nerve impulses that travel from one brain cell to another brain cell, go at about 250 miles an hour. So we're really, the speed between the nerve cells is really tremendous. And that is actually something that changes dramatically during development, and I'll talk about that as well. And it has direct implications of how the young brain sees and processes information compared to how the adult brain processes information. So this is the organ we're talking about. It's basically, that's a picture of the brain. And what you immediately recognize, you have these, basically these windings here, and then you have these valleys or these fissures that separate these windings. So why is that? It turns out that the way the brain organizes the nerve cells is that most of the nerve cells live on the surface of the brain. And in order to pack as many nerve cells into the brain as possible, the brain had to develop a strategy, or the biology had to develop a strategy to increase surface volume or surface area. And basically that's what caused the brain to develop into these windings and into these, basically into these valleys in between the windings. So that's called the gyri and the sulci, and we'll talk about that in just a moment. But let's focus in a little bit on this yellow box here and see what the brain looks like as we're focusing in. So as we're focusing in on the brain, what we're seeing is these, what's called the gyri or gyrus, and the sulcus, which is basically these fissures. And the whole area that is basically on the outside of the brain is called the cortex. And as you can see, there is sort of an area here that's a little darker than the area here in the middle. And the area on the outside is called the gray matter, and it's called the gray matter for a very simple reason. When the anatomists first studied the brain and they looked at it, it looked gray. And then if they look into the brain, it looks white. And it looks white inside because most of the inside, most of this here is cabling. It's actually isolated cabling. And it's the isolated cabling, and the isolation is actually is the sheets around the nerve cells that are mostly fat, and that's mostly white. So that's why this is called the white matter, and this is called the gray matter. And again, this is important because this changes dramatically during adolescence, and we'll talk about that in just a moment. Again, let's focus a little bit more on what's happening inside this yellow box, and we'll go a little more in detail. So here's the next level. So basically, as we're focusing in, it turns out that the gray matter, this area here, is organized in what's called columns. So the columns are basically the way that the nerve cells arrange themselves. And it's usually what happens is that information travels from the very edge of the brain all the way to the bottom, which is where the cabling is. And within those columns are various types of nerve cells or brain cells. And these brain cells obviously make connections, and I'll talk about the connections in just a moment. But so these columns are organized in layers. So just like a layer cake, there's several layers. And most of the brain surface has 6 layers, 1 through 6. There's some other brain areas that only have 5 layers. And that has really some significance, and in some of the other lectures we'll actually talk about this. This picture here was generated by one of our neuroscientists here at LIBR, Dr. Kyle Simmons. So let's further focus in on the actual nerve cells. So let's go down one more level. So that's a picture of a nerve cells. And nerve cells are really incredibly fascinating cells in the body. There really are 4 parts that you need to know about for the brain cells. And these 4 parts are important because these 4 parts change, again, during development. So the first part is the cell body. The cell body is basically, you have the nucleus. The nucleus has all the genetic material. The genetic material tells you what kinds of proteins and what kinds of messengers to produce. It has what's called the dendrites. The dendrites are basically the input to the nerve cell. It's where impulses coming in. And then you have, on the other side, you have what's called the axon. The axon is the output. And what you can see here is that around the axon, so the axon is this yellow cable here, and around these axons you have these sheets. These are they're called myelin sheets. And what that does, it helps to speed up the information transport. Because what happens is you get impulses coming into the dendrites that they basically travel through the nerve cell, and then they travel along the axon. And normally, if you don't have these sheets, they have to travel really little step by little step. But when you have these sheets, they can actually jump. They jump from across these insulation cables to these open areas in between, and it makes the transport much faster. So the impulses, the electrical impulses, travel along the axon, and then they end up at what's called the synapse. The synapse is the connection of one cell with another cell. And the synapse is really fascinating because in the nerve cell, information is encoded in electrical signals. By the way, the brain generates about 12 to 25 watts of energy. So if you could harvest the energy of one brain, you can almost light a light bulb. But basically what happens is that this electrical energy is transmitted into chemical energy, and the chemical jump over from one nerve cell to another and start up another electrical reaction. And that's what's happening at the synapse. There's a lot of things that happen there, and there's a lot of interesting insights that we'll talk about probably in one of the subsequent talks. But again, in order to kind of understand development, you have to understand that there are the cells, there are the axons, there are the sheets, there are the synapses. The synapses make the connections. Okay, so now you have the basic building blocks. Now we can talk about what happens during growth and development. Okay, so here's a graph that is actually quite surprising. What you see on this axis, the x-axis, you see the age in years. So basically from birth all the way to age 24. And on this axis, the y-axis, you see percentage of the adult brain size. And one thing is immediately obvious, is that the brain really grows really rapidly early on. By the time that the baby or the child is 2 years of age, it basically has already had its brain is already 80% of its final size, which is really unusual. And we'll talk about this in comparison in just a moment. So you have this rapid growth over the first 2 to 3 years, and then you basically have sort of a slow asymptotic or a slow growth to finally about 100%, which is usually reached at about 18 years of age. So that's basically when the brain has reaches full size. And so if you compare that to other organs, so here again we have the age on this axis, and you have basically the weight, which is another way of showing size. You see the brain reaches its size fairly quickly, and if you compare that to the heart and the general body growth, you can see that most of the other organs grow much more linearly and much more similarly to the body. So the brain is actually kind of an early comer. So you may ask yourself, now how the hell is that possible? Well, let me show you the next picture. So the next picture shows actually in scale the size of the head relative to size of the body. And this almost reminds me of Aliens or something. Yeah. Because essentially, I mean, look at this. I mean, it's a huge head. Right? I mean, and what you see is that relatively speaking, over time, the head gets smaller, right? The body gets bigger and the head gets smaller. It also, it's important to actually understand this, because a child, for a child to have good head control is actually really difficult, because it really, compared to its other muscles, it really has to develop some strong neck muscles to be able to really keep the head stable. It also, and this is of course another unfortunate outcome, the head is extremely sensitive to rapid motions, right, to shaking. And you can now understand that, you know, particularly in babies, if you shake a baby, you really exert a tremendous amount of force on this head. And really makes it, you know, can have tremendously negative consequences. But so you basically, your brain is getting relatively smaller to the rest of your body. So this is sort of the outside. This is basically what the brain and what the head and the brain looks like compared to the rest of the body. So let's now talk about the nerve cells, the connections, and the development. Okay, let's start again with our nerve cell. Remember, we talked about the dendrites, the cell body, the axon, and the synapses. So let's talk about what happens with all of these components. So this is a picture that shows essentially is a visualization of a column of brain tissue. Remember that the gray matter was organized in columns. And what is done here, you stain, meaning you mark the nerve cells in black, and everything else around it, which is mostly connections and other cells, is sort of shown here in red and orange. And so across here, you start with the newborn, and you look all the way to adults. So basically at the newborn, you see a couple of cells, and again, they're organized in these columns from top to bottom. And then as you get into early life, you see more of these nerve cells popping up. So you realize, okay, there's still cells that are growing. And as you get to 9 months, it really starts to look a little crowded here. And then even more so at the age of 2 years. So now you have a lot of cells, and you have a lot of these connections, a lot of these dendrites and axons floating around in the column. And then you notice something really interesting. So when you look at the adult brain, it looks like that there are fewer nerve cells, and there are fewer of these axons around. And that is the first very important thing about development. It actually turns out that at first we gain nerve cells and then we'll lose them. And the loss is actually really important. It's really important that we lose nerve cells. Because if we don't lose nerve cells, and we know this now from a number of different studies, that the kids that don't lose nerve cells often have autistic traits. So autism goes along with not pruning enough synapses, not getting rid of enough nerve cells. So that's something important to remember. So this is basically the progression. Now the next picture shows that in a little more detail. So here we start actually before birth. Here's the newborn, 3 months, 6 months, 2 years, 4 years, and 6 years. And what's basically happening is that you have a period, particularly over the first 2 years, that is dominated by synapse formation. Again, the synapse is the connection between the brain cells. And then you have what's called synaptic pruning, or synapse pruning. That is when actually some of the nerve cells get stripped. So how does that happen? So the way to think about this is that the way that these synapses are formed and not formed is that the nerve cell reaches out. It's sort of like, the analogy that I came up with may not be the best analogy. Say you go to a football game, and you watch the football team, and you get really excited for your team, and you scream and yell, and out of a sudden next to you, there's another person that screams and yells, just when you scream and yell. And you look at the person and you say, hey, he's voting for the same team. And so you say, oh, how are you doing? And it's pretty soon you in a conversation, and you made a buddy. And that's essentially how it also works with nerve cells. So what happens is the nerve cells are reaching out. They're seeing that another nerve cell processes information that's similar to what this nerve cell is processing, and it makes a connection. And that helps to better process the information. And then there are other nerve cells. So now again, coming back to the sports analogy. So then maybe there are other people that vote for the other team, right? I mean, they scream whenever you don't scream. They're upset when you're happy. And so pretty soon you say, I don't want to hang around these people. And, you kind of go in the other direction. And that's exactly also what happens with the nerve cells. So nerve cells that don't fire together or that don't process the same information start to kind of separate. That's essentially what's happening. And that is at the basis of synaptic formation and synaptic pruning. So now let's take a look again back at the gray matter. So you have the gray matter that changes, again, dramatically during development. Gray matter was essentially this, the column of the nerve cells that I showed you just a minute ago. So here is a bunch of graphs that depict what happens during development. In fact, these graphs were generated by one of our collaborators of our new study, Dr. Jay Geed. And I'll walk you through each of these graphs and help explain what's happening here. First thing you see, again, on this axis, you see the age of the child or the young adult or adolescent. So we're looking at a range from 6 to about 20 years. And then you have different components of the brain. And on this axis, you have the volume. So what happens is, and then you have blue and red. And I hate to say it, but let me just preface this. Bigger is not always better. But it turns out that the male brain is about 10% bigger than the female brain. But again, it doesn't mean that there's any... but it turns out that the blue is males, the red is females. But you see that the overall total brain volume, as we talked about, stays pretty constant during this period. But what happened is this gray matter significantly goes down particularly during the adolescent phase. And that is true for both the males and for the females. So what happens is you lose gray matter. What is gray matter? It's cells. It's the brain cells that we talked about that are in those columns. So we lose cells during adolescence. At the same time, we gain white matter. And the white matter, again, is the cabling. And remember, the cabling is mostly this insulation cabling. So the one that makes it so much faster to get information across. And what happens here is that information transport gets incredibly more effective. And I'll show you a picture to kind of visualize that in a moment. And at the same time, you see a little bit of change so there's one part of the brain I actually didn't talk about today. There's actually fluid-filled spaces in the brain. They're called cerebral spinal fluid, and they're called ventricles. There are different types of ventricles. They stabilize the brain, sort of like a fish bladder, and they change very little. They stay pretty constant. So we see total brain changes little, gray matter goes down, white matter goes up, and this one stays pretty much the same. And then we can look at different parts of the brain, and the cortex definitely goes down, and then some other parts that kind of stay pretty stable. And I'll show a couple of other brain parts in just a moment. But that's really important to know, that during adolescence, there's a rapid and significant decline of gray matter. Okay, here's another picture of this from another group. And basically what this shows is for different brain regions, again, on this axis you see age, on this axis you see actually volume. And what you can see is that for the most part, most of these areas, there's a decline from early age to early adulthood. And this is basically very similar. And I show these two graphs to tell you that this is no longer in question. We know that that is happening. We know that that's an essential part of development. Now this is a depiction of this and shows one more piece of information. This was actually generated again by one of our collaborators, Dr. Jay Geed. And what this shows is how the cortex, remember that's the surface of the brain, how the cortex gets thinner with age. And so what it shows is in color code how much it thins out at different ages. And what you can see is that the thinning starts in different places first, like here in the back of the brain, on the top of the brain, and a little part in the very front of the brain. And then it propagates, it basically gets more so, and eventually it reaches its adulthood. And this is also important because what that shows is that the thinning is not uniform, that certain parts of the brain thin before others. And I'll get back to that later on when I talk about hot and cold thinking. But this is, again, it's very well established now. However, one of the things that we don't understand is what drives this process and what is the individual variability. We know that this happens on average, but we don't know how that happens for an individual person and what contributes to this. So this is something that we really need to know a lot more about. Okay, so in order to give you an idea how that matters, what I've done is I've taken a picture and coded it in a way that a child sees the picture or processes that information, and a way an adult sees that picture. So here's a picture of a nature scene. And you can see clearly, you can kind of make out what it is, a mountain, there's a lake here, there's a little bit green. But this is the picture of what it actually looks like. And so the way to think about this is that the child really sees this and processes the information on this level, whereas an adult is able to process information a lot more efficiently and therefore is able to detect a lot more details in whatever the information is that comes in. And that is really the result of this pruning, of this getting rid of these of the nerve cells and making the connections more efficient. So next I'm going to talk about critical periods. Yeah?
Isn't it also that as an adult you have a larger inventory?
Yes. Yeah, that's another good point, that as we age, and this is actually something that it'll become very important here, we have stored information and memory that we bring online to help us better understand what we actually are seeing. That's a very important point. So in addition to seeing things more clearly, we also have a lot more context to put these things in. Yeah, it's very, very good, very important. Okay, so now let's talk about critical periods. So what are critical periods? Well, we have known for some time that if you look at development over time, and if you look at plasticity, and the plasticity might be a measure of how quickly you learn something, how well you learn something, that there are certain times when plasticity for certain functions peak and when they go down again. Now, I want to actually say something here, because I think that there has been a misunderstanding. There's a lot of folklore that most of the plasticity happens during the first 3 years of life, and that once the 3 years are over, basically you're done. That's not true. We know that the brain is a lot more plastic, a lot more dynamic than that. We know, in fact, that certain functions, certain higher cortical functions, don't even come online and don't even get optimized until in your middle age. 30, 40 years old. So we need to really think of these critical periods as happening throughout life. That the brain opens a door, lets you learn, and then it closes it again. So now, of course, how does that door get open? So I want to talk a little bit about that. Okay, so here's another picture of this. Again, you see age on this axis. And basically you see that seeing and hearing, which is processed by the visual and auditory cortex, happens really early on in the first few months. And it is true for some of these functions that if you don't expose the child with specific rich stimuli during these early periods, it makes it harder for the brain to process that information. So you start seeing and hearing has sort of a peak early on. Then you have language, which comes online around the first year of age. And then you have these higher cognitive functions. But again, this actually extends, and we have a lot more knowledge that extends into adulthood. And it's essentially another formulation of this picture, just with a lot more information. So how does that happen? So let me talk a little bit about this. So essentially, this has been studied mostly for these very early periods, but there's enough evidence now that some of the same processes, some of the same molecules are important for these other critical periods as well. So there's a lot of weird words on this slide, and I basically want you to not be too concerned with most of them. I'll mention one, because I'll bring that one molecule back a couple of times. So basically what happens is there are certain molecules that are important for keeping the door shut, because sometimes you don't want the brain to be plastic. So there's certain things that, certain molecules that keep the connections in check. And then there are others that then start to open up the door. And then there are still others that make it really important to keep the door open. And then there finally are, again, another set of molecules that put on the brakes that said, okay, now the door is shut. And so, again, what we're trying to figure out, and part of what we're doing here, is how that relates to things like depression, anxiety. But one of the interesting things I want to point out to you is this little guy here called BDNF. It's called brain-derived neurotropic factor. Don't have to remember it, just call it BDNF. This will become actually quite important in a number of slides, so that's why I'm highlighting it. And this one is definitely one that's important for opening a door and actually keeping the door open as well. So basically you could say opening the door, opening the window for new development. Here is another way of showing this. Again, it's a whole cascade of molecules that basically open the door, keep the door open, and closing the door. And again, here's our friend, the BDNF, and it's actually here too. So it opens it and keeps it open. And then there's other things that closes it. So why do I keep harping on this BDNF thing? So you can see him right here. So here's a totally different picture. This is from a totally different field. This actually is a summary, and again, there's a ton of information on here, so most of which I wanted you just to ignore. But this is a recent review of all the chemicals that change when you do regular vigorous exercise. It's a whole host of things that change. But guess what you see there? There's our little friend, the BDNF. So what that tells you is that exercise, which tends to really boost BDNF, it's been very well established, is the molecule that also opens the door for new development and keeps the door open for new development. So if there's one thing, without reaching too far, that I can definitely say that is a natural way of keeping your child's door, opening your child's door and keep it open, is definitely exercise. There's no question about this. Because chemically, it really promotes the growth, the connections of synapses between the brain cells. So that's my way of showing this. But it goes even beyond that. Here is a sort of a summary, and again, without going into too much details. Not only is the BDNF actually important for opening the door and keeping it open in development, it actually is also, and you can see it here again, it's also important for reducing depression. It's one of the things that actually has been shown in a number of animal and also human studies, that exercise is probably the best natural antidepressant that we have. So now let me talk about stress and development, because I think it's really it's one of the things that is really important to understand as well. So what is the stress system? Let me start again very much from scratch. The stress system really consists of a few parts of the brain. First is what's called the hypothalamus. It's a part of the brain that sits really at the bottom in the middle, and what it does, there are particular nerve cells that squirt out substances called corticotropin-releasing hormone that then gets signals to what's called the pituitary gland. Pituitary gland is another part of the stress system. And when the pituitary gland gets this CRH, this short term, then this in turn squirts out what's called ACTH, or adrenocorticotropic hormone. Really long. I mean, it's all the scientists had nothing else to do. It actually is really interesting. Let me just tell you, this is old school science. This was sort of 1950, 1960 scientists. They were all Latin freaks, right? So they would name everything in Latin. Now you have the new molecular biology science, and they have all these funky names. I can't right now, but they basically name everything funny. So, you know, all the new molecular substances have funny names. So the next generation will come up with something altogether different. But this is basically the CRH that basically stimulates the ACTH, and then that goes to the adrenal. It goes to the blood and gets to the adrenal gland. The adrenal gland sits right on top of your kidneys. It sits right on top of the kidneys, and then it squirts out cortisol. And we all know, maybe we all know, cortisol is the stress hormone. So now, why is it so damn complicated? You know, why are there so many different pieces in here? There's so many different pieces in here because the stress response needs to be highly regulated. So here's my local example. I like to bike ride. And I bike ride in the vicinity of Tulsa. Maybe some of you bike ride too. I know some of you bike ride too. And sometimes when you go through, you know, some of the rural areas, you might come around the curve, and all of a sudden you see a dog. And the first thing that the dog looks at you, and for some reason, it's really, dogs just kind of love bike riders. and they really, it's like something goes off and they start running, and you have to make a decision. How can I out-pedal the dog? But obviously what happens during that period of time, you come around the curve, you see the dog, your hypothalamus goes, oh my God, maybe other words too. It basically expresses the CRH, it gets to the pituitary sends it to the adrenal, and adrenal makes you like really bike hard, right? So, through the cortisol. But then, you bike away, the dog is gone, and now you have to get back into a regular state of affairs. So you have to shut that system off really quickly too. And that is why you have these multiple circuits. There's a lot of feedback loops that is built in here. Because you want it to come off very quickly, and you want to shut it off very quickly. That's basically the stress system. Okay, so now how does that change with different types of developments? Okay, so here's actually a summary of what happens when you are chronically exposed to stress. So now, forget the dog, but think about a chronic stressful situation that doesn't go away and constantly challenges the brain, stress. What happens is that you have basically, you exhaust the system, is basically what happens. You have these, because you have chronic stress, you have much less sensitive stress response. And the stress response doesn't shut off. It constantly signals. So it would be as if I were biking all day long thinking there will be a dog around the next corner. And I would be stressed the whole time. And that is what happens. And we know when that happens with lots of, that these kinds of changes happen with a lot of early life adversity. And as a consequence, you have this overactive, what's called the HPA axis, hypothalamic pituitary adrenal axis, which then kind of sets off anxiety traits, and which actually then can also result in depression, and even in this particular case, they were talking about even suicides. So the point here is acute stress is something that the brain can deal with really well. It can, and it has a lot of different systems built in. But chronic stress is a whole, together different matter. And so what we now know, here's another example of this, that basically what happens is with chronic stress, with chronic early stress, you are making the connection between the systems that's supposed to keep the threat system in check weaker. You weaken this connection, which is one part, and we'll talk about this connection at another lecture. But the point here is your system becomes less and less able to adjust to stress when it's there and when it's not there. Okay, the final topic. I want to talk a little bit about hot and cold thinking and feeling. And this is particularly relevant to adolescent development. Okay, so what is hot and cold thinking? So cold thinking or cold cognition is really sort of what we think of normally of the thinking process. It's memory, it's attention, working memory is a form of memory, sensory process like hearing, seeing, different language. Those are all what's called cold cognition. And then you have hot cognition. That's basically processing of emotion. If I look at somebody's face and I decode emotion, if I try to differentiate one emotion from another emotion, if I hear somebody's voice and I'm trying to think, is he angry or is he happy? If I'm communicating my intentions to others, or if I have empathy, if I try to feel what the other person is feeling. All of that's part of this hot cognition or hot thinking. And the point is that these two systems are supported by different parts of the brain, and that these different parts of the brain develop at different times. So what happens is that you have the more advanced part of the brains, the frontal part of the brains, that are very important for this cold cognition, But you have these more basic part of the brain that develop quite early, like the threat detection system and some reward and punishment systems that develop early. And they are usually quick. You may have heard of the quick and slow thinking. And in many ways, the hot and cold cognition is sort of a similar an analogy to this. The cold thinking is more deliberative, it's effortful, it takes some time, whereas the hot thinking is quick, it's very intense, emotionally intense, and there has to be basically some negotiation between those two. But as I showed you earlier, remember when I showed you those pictures, those colorful pictures of how the cortex developed, I showed you that some of the cortex develops later than other parts of the cortex. And in particular, this part actually it develops one of the latest. It's called the dorsolateral prefrontal cortex, long word, you don't really have to remember, but it's part of the frontal brain. And part of the frontal brain really doesn't come online fully until about 18 to 25 years of age. So you basically have this part, which is sort of the cold thinker, basically the boss that tells you when to do what, and you have here the urges that come on, and that may have very strong intentions, and the boss may not be fully in the office yet. And so, as a consequence, it actually, you can show that some of these cold cognitive deliberative thinking are actually developed fairly late. So what I'm showing you here is a bunch of graphs, and I'm only going to go through two of them to make a point of how these things develop. So let me show you the first one here. So the first one is actually, it's a very devious task. So the way to think about it is you're looking at a screen way in front of you. And all of a sudden, something flashes on the right-hand side of the screen. And what's your first response? You want to look at the right side. What the hell is over on the right side? But you're being told beforehand that when you see a flash, because you see it on the periphery of your vision, when you see a flash on the right side, I want you to look at the left side. That's pretty diabolical, isn't it? So basically, you have to inhibit your urge to look at the right side and look at the left side. And that's one way of how you can test how well developed this executive system is, sort of the boss in the brain. And what you can see here, again, age is on the x-axis, and then on the y-axis, you basically see the errors that kids make. And you can see that it's dramatic, that it really takes, kids of 8 years of age make a lot of mistakes, and it takes them a long time to overcome this urge. And then only later, as they grow up further, are they able to overcome this. And again, this is important because it shows you that the inhibitory system, what we call the inhibitory system, is really not fully developed. And it's really, this only shows it to 12. But you can show, and here's a couple of other graphs, that this actually takes quite a bit of time to develop, even into early adulthood. So this is another example. In this particular case, again, it's sort of one of those tasks that somebody came up with, and once you do it, you really know how, experience how hard it is. What you basically have to do is you see the word green that's written in green ink. And you're being told you have to name the ink, not the word. So in this particular case, that's easy, right? Because it's green, and you say green. But then you see green in red ink. And so what's your first response? You want to say green again, but no, you have to say red. And you basically can show that this is, you have to overcome. It takes quite a bit of sort of the executive. And you can see over time, this again is how difficult it is for the kids to do that. As the kids age, it becomes easier, but it's clearly something that develops over time. So what's the consequence of that? So the way people have been thinking about this is that you have, as part of adolescence, you have these two systems, this hot thinking and feeling system, and this cold thinking and feeling system. And in essence, as the brain maturates, or as the brain develops, you have the urge-based system that essentially overcomes, for the most part, or is stronger than the cold thinking system. And only later, as you enter adulthood, are you able to better negotiate. It's sort of like playing football with somebody who is 2 sizes bigger than you, and you basically have to work really hard to make any advances. And on top of that, of course, what happens during adolescence, you have enormous hormonal boost, which are sort of the bottom at the bottom of this kind of these emotion processing areas that are kind of coming online. So you can imagine that it is really hard for an adolescent to overcome some of those hard thinking and feeling urges. And part of what, in terms of practical, in terms of practical applications, if you understand that basically during adolescence you have these strong limbic urges that come on, and you have the relative weak prefrontal. In a sense, as a teacher or as a parent, what you can do is provide external help in helping with this, right? You can basically lend your brain to the adolescent brain. Of course, the adolescent has to let you, which sometimes is not too, it's a challenge by itself too. But the point is, it's not that the adolescent doesn't want to engage this. It's just that it's much harder for that person to engage that part of the brain. And that is all due to the development. It's due to the fact that the neurons have not yet fully matured, that the brain cells have not made all the connections, and that the connections are not yet that efficient. So what are the take-home points? So let me wrap up. So what do we know? What we know is that the brain undergoes tremendous development from birth through adulthood. And again, I want to emphasize, yes, some of the basic functions develop fairly early, but some of the more advanced functions really develop throughout life. And that goes along with this point. The brain is a lot more adaptive than what we previously thought. And I think that's a really optimistic perspective. We really need to think of the brain as something that we can, at any age really, we can challenge, we can open doors. There are critical periods when the brain is able to learn particular things. I showed you about what opens the door, what keeps the door open, and what closes the door. I also showed you that one factor that contributes to that is this BDNF. And we know that a vigorous exercise definitely helps to open and keep the door open. So that's this part. The exercise can open the door for the brain to learn and keep the door open. This is also an important point. Severe and chronic stress has profound and long-lasting effects on the brain. We know that early lifehood stress, because it changes the stress system, and that these changes last. So again, from a practical perspective, we need to appreciate that some of the youngsters that we encountered may have had experience of early life stress and may even still encounter stress. And so the way to think about it, again, I'm coming back to my bicycle analogy. You and I may see the dog, may get all upset, and then we go around the corner and the dog is gone, and basically it's out of our mind. You know, we're back to sort of normal biking. And a child or an adolescent that has gone through early life events, for that child or that adolescent, that dog is still around an hour after the dog really was there. See? So you have to appreciate that. And that's something that we are just beginning to figure out, how do we reverse those effects? Because of course that's at the heart of some of the things that we're interested in. And so then finally there's a critical push and pull of hot and cold thinking and feeling during adolescence. And this is again the consequences of the development, that the executive, the kind of cold thinking part of the brain is not fully developed yet. You have these hot urges, emotion processing that are important, and you're trying to negotiate them at a time when lots of lots of things are happening. And of course, for our study that we're trying to start this year, it's exactly that period that we're interested in. What happens in that push and pull period? What helps the adolescent to negotiate that push-pull? And how do we influence that push-pull so that the person has a more successful life? Because as you can imagine, the adolescent period is incredibly important for adult development, right? That's a lot of stages for what we will be as adults. So those are the main points that I wanted to leave you with today. And I want to thank you for coming in. I also want to say that this would not have been possible with the support that we get from the William K. Warren Foundation. And a lot of this was really collaborative work together with Amanda Morris, Kyle Simmons, Teresa Victor, Florence Breslin, and Colleen McCullum. So that is all I wanted to tell you about today, and I'm open for questions. Thank you. Yeah. So part of that has to do with, so the cortical thinning is a direct consequence of two things. One is that fewer gray cells and fewer connections. We know that that with two, it's sort of like when you're in a stadium and you have a crowd and the crowd hasn't yet decided who to vote for. You have a lot of chatter. But so there's a lot of noise, but there's not really coherent information processing. But once you kind of figure out, okay, who to vote for, who to root for, there's a lot more coherent activity. And it's the coherence that is really helped by the thinning, so that there isn't sort of random chatter going on.
I know you've been interested in studying and have studied addictions and substance use. I wonder if we know the effect, because kids don't wait to medicate themselves until they finish developing. A lot of kids using drugs, alcohol,
Yeah.
14, 15. Do you have a sense of what that's doing to that trajectory?
Well, that is in essence one of the key questions we're trying to answer with this study that we're starting out with the ABCD study. Because yes, that's exactly so the question is essentially what happens to that change, to that trajectory when the child or the adolescent starts using drugs, and how is it altered by drugs? The truth of the matter, we don't fully know. There have been some studies with a small number of kids. There is some evidence that, in particular, interestingly, alcohol has some fairly profound effect on the connectivity between different brain areas. But we really don't know because most of the time these studies look at kids that have already been exposed to alcohol. So there have been really no studies that have looked at looking forward from before the kid was there using and when it started using. And that's very important because even though I showed you all these beautiful graphs and how this thins out, and this is true for the average, but we know there's tremendous variability. So we really have to understand that for an individual child and not for just the group. So that's really one of the key questions we're still trying to address. Mm-hmm. Ecstasy.
And how that helps people overcome.
Yeah.
I mean, is that true?
Yeah. So that's it. So the question is about MDMA or ecstasy, and some people have used it in psychotherapy even to help with closeness. So MDMA is it's what's called a designer drug. It was created about 70 years ago at first. And it has unique properties. It works on two different chemical systems. One is called the dopamine system, and the other one is called the serotonin system. And the prominent feeling of what people experience with MDMA is a sense of empathy with others and feeling close to others. And so it has been used in psychotherapy to actually increase that empathy. There is, it's a very controversial topic for the following reason. There have been some studies in monkeys, and there are some studies in humans, although they're people on both sides, that suggest that MDMA may be neurotoxic, may actually destroy neurons. That has been highly contested, but some people feel very strongly about this. So the short answer of this is that it certainly has these empathic abilities, or engenders these empathic abilities, and in that function, it has been helpful for psychotherapy. The judgment of whether it's bad for the brain is still out. As a consequence, the FDA has made it what's called a Schedule I drug, which means that you cannot prescribe it for anybody, and it's very difficult to study. But I think that there is, at least in that class of drugs, there may be a function for enhancing psychotherapy in the future. And that may be something we can talk about in upcoming lectures, which is how do we boost psychotherapy, and how do we make psychotherapy more effective? How do we engage people? And there may be a role for something like MDMA. Maybe not MDMA, probably not because of the legal issues. But it's intriguing, and it's been well replicated. So the effect is definitely there. MDMA tends to increase sort of emotional closeness to others. Mm-hmm. Thanks. For inhibitory development? Yeah. Yeah, so that's a really good question. So the question is really similar to with hearing and seeing and other functions. Is there a sensitive period of inhibition when you basically when the brain is able to better develop inhibition than during other times? And as far as we know, as part of executive function, inhibition develops sort of during late adolescence, early adulthood. Probably not done until about the mid-20s. But people have really not studied sort of the critical periods of inhibition. It'd be very fairly straightforward actually to do so. But as part of executive function, it probably doesn't develop fully until about late adolescence, early adulthood. Again, related to the brain areas that are fully maturing during that period of time. Inhibition, as it relates to the cortex, it happens a lot in what's called the inferior frontal gyrus. It's a part of the lower part of the brain in the front that is important for kind of the break. And that function and that part of the brain really doesn't mature until sort of the late adolescence, early adulthood. But to my knowledge, there hasn't been really a clear study of critical periods of opening and closing as it relates to inhibition.
Could you address ADHD in terms of brain development?
Yeah, so that's another interesting question. And we'll have one of the upcoming lectures. We'll definitely talk about ADHD. So let me just say a little bit about ADHD. So ADHD, or adult attention deficit hyperactivity disorder. A lot of people think of it as an attentional disorder. It actually is much more what's called an executive functioning disorder. And what that means is that the planning part of the brain has real difficulties coming online. And what's interesting really is that it's thought now that the hyperactivity part may actually be a way of compensating the attentional or executive dysfunction. Because what we know, and again, saw the BDNF as one kind of the hyperactivity, certainly squirts out more BDNF, but it also squirts out more dopamine, and dopamine is a chemical that's very important for regulating executive function. We know that ADHD is very genetic, that it's about 80% of, upwards of 80% of it is genetic. It comes both in an overt kind of hyperactive state, but it also comes in much less overt, sort of pure executive attentional dysfunction state. There had been the idea that most of it, that kids grow out of it in a sense, and that's true to some degree, and you can understand that because what we understand from a brain development perspective, a lot of it is actually related to frontal functioning. And as we know, frontal functioning matures late. It matures in the late adolescence, early adulthood. And it is true that a significant number of people, probably about half, will be able to have better what's called executive control as adults. But there will still be about half of the adults that were as kids ADHD will still have ADHD or executive-related symptoms. The difficulty with ADHD, and so now so obviously the standard treatment for ADHD is stimulants. And stimulants, what they basically do, they raise dopaminergic levels. That's basically how we understand that they're helpful. We actually do know, on a side note, children with ADHD are at high risk to engage in drug use behavior. And that the way we understand that is that they're looking for ways of calming themselves down, and they come across different drugs and then they use them. And we also know that kids who are treated are at lower risk. So in some ways, the treatment actually has some preventative function. We do know that long-term use of stimulants, as far as we know, and people have looked at that with brain development, don't have adverse effects on brain development. So as a psychiatrist, I would say, if you have clear-cut ADHD symptoms, the treatment with stimulants is definitely something that is useful. The problem is that there isn't a clear test for ADHD. It's not like, and that's, again, something that we're trying to develop at LIBR, not particularly for ADHD, but for other diseases. If you think of psychiatric diseases, we don't have, like a finger stick, like when you have diabetes. You get a finger stick, you get a high blood sugar, you have diabetes. We don't have that for psychiatric diseases. And the problem with ADHD is there are lots of things that can appear like ADHD, and sometimes it may just be a normal extent of development. So it can be a little tricky, but for the clear-cut cases or clear-cut examples of ADHD, early treatment, continued treatment, and the treatment should not just include medication. It should clearly include helping the child developing these executive strategies. How to gain better executive control. Physical activity might actually be really important in that sense because, again, it helps the brain to open up the developmental door and can be extremely useful there. But I don't know whether, I gave a lot of different bits and pieces about ADHD, but I don't know whether that addressed some of your concerns.
Vigorous physical activity, for example, I think there can be a big difference between having a class do vigorous 5 minutes, really vigorous, not standing around like they do in gym class. So
Yeah.
Instead of having a whole period during the school day for the gym, just have every hour 5 minutes of really intense.
Yeah.
Would that make any
I think that
difference?
Yes, particularly for BDNF, we know that short periods of intense exercise actually can raise the BDNF levels relatively quickly, and are much more effective than sort of low-grade exercise. So one could conceivably see that, instead of a 5-minute break sitting at the desk, having everybody running around, for 5 minutes. And in a sense, beyond just the BDNF, we know that there are enormous benefits of this, just physically for the body and so on and so forth. So the studies haven't been done, but all of the biochemical pathways point that would help with learning behavior.
Just for 5 minutes.
Mm-hmm. Yeah, very short, vigorous exercise. Yeah. Mm-hmm. Yeah.
I don't know if you can be able to answer this one, but from a neuroscience viewpoint,
Uh-huh.
what's the difference between recall and thinking? How would a neuroscientist describe thinking, the mechanism of it, versus the mechanism of recall?
Well, so let me kind of probe a little bit. So you're trying to separate recall, which is essentially going back in time and remembering a piece of an episodic event, something that happened in your life, and then engaging in that. So we know that episodic memory is actually a very particular kind of memory that is stored in parts of the brain that is also involved in the thinking part. So when you basically, when you say, okay, so 20 years ago when I met Joe for the first time, what you're doing is you're engaging the frontal part of the brain to kind of pull a storage in of a memory, and then you bring that back to the frontal part of the brain. That's what's called working memory. Working memory or scratch pad memory is essentially what we're doing when we're thinking. Because basically what the brain does a lot of things we have no idea that it's doing, right? In other words, when I walk around here, I keep my balance, I look at people, I get a sense of what's happening, I feel hungry or not hungry, or thirsty, not thirsty, all that the brain is doing, all that. Except that most of the time, I'm not paying attention to that at all. It's not in my scratch pad memory. Recall is a function of bringing these memories into scratch pad. Thinking, and so let me just make an interpretation, you can correct me when wrong, is in a way sort of associating, thinking forward, thinking what might be happening. Is that what you're thinking as thinking?
I have a hypothesis
Mm-hmm.
that there is no difference,
Mm-hmm. You're probably right.
and that all we're doing is we habituate. So if you use it often enough, you're going to access it often enough,
Mm-hmm.
and then you think you're thinking, but you're really not thinking. It's just it's not a continuous process.
Mm-hmm.
Jump.
Yeah.
And we interpret it as thinking because it's happening so doggone fast. And so we fool ourselves in thinking we're thinking, but all we're doing is we're recalling things that we have habituated because we have such ready access to them.
Yeah, it's certainly true that many of the thoughts that we have on any given point in time, it's actually, so what's really interesting, so one of the things that we're particularly interested in, actually, we've studied, is what's called a resting state imaging, or fMRI, resting state imaging. What that basically is you put a person in a big MRI machine, and we'll talk more about what MRI is and so on. You put them in a big MRI machine, you have them lay there, and you have them do nothing. Well, now, how many people can really do nothing? So what are you going to do? Is you're going to think about, what I have for lunch today, and what I'm going to do tomorrow. And we do know when people engage in that, we have particular parts of the brain that really go to town, and you're really active. Some of the same areas that go on when we're thinking. Although there is one big difference. When I asked you to, for example, I give you a task let me give you two tasks. One task is I give you the number 100, and I want you to subtract number 7. And I want you to do it until I say stop. And so, you do that, and that's sort of the kind of the classic working memory task, right? And another may be, I want you to think about all the things that that are white, say, and you start thinking, okay, snow, ice, polar bears, whatever not. So when you engage in that kind of processing, most of the time you're using what's called the lateral part of your brain. For this kind of more self-oriented brain, you use actually more the middle part of the brain. So at least when we put people in the MRI machine, we do see a difference between sort of this kind of habitual, what you call habitual episodic memory, and sort of when I really task you to do something that that maybe is something you haven't done, or something that you don't do as often. And then you engage more these lateral parts of the brain. So to some degree there is a little bit of a difference. But it is true that, see, what's interesting is our language developed long before we have all these tools and techniques available, right? I mean, we think in terms of psychology oftentimes, what's an emotion? So by the way, everybody knows now Facebook has 5 emotions. So pretty soon, that'll be the end. 5 emotions will be it, right? So we think of it in psychological terms, but actually the way the brain processes it may be quite different. And the reason why that's important is because if we really figure out how the brain processes it, what we found so far, that may actually tell us a lot more about how a person will be doing tomorrow or a month from now than sort of the psychological approach. How do I feel right now? People are actually not very good in telling us really how what they're saying is not necessarily helping us. And that's also, again, coming back to why we need these brain imaging studies. We really need to understand the development and how thinking and feeling develops, not just by the way psychology tells us, but also by the way the brain tells us. Yeah. Mm-hmm.
And I asked him, which part of the brain tells you not to do that thing? And he didn't draw that.
Mm-hmm.
So, in children as young as that, how does this inhibition system work?
Yeah.
Because they're always being told, don't do this, don't do that.
Yeah.
And they will not do it for a period of time, and then they will turn around and get it back.
Yeah. Well, so that is a great question. Is because in the early stages of development, those inhibitory parts of the brain are not yet fully online, and yet, obviously part of what parents do is provide their brains to their kids' brains, right? So, and we can learn to do a lot of not doing by habitual learning, right? And just like we learn how to brush our teeth every morning, or how to take a shower, or how to take the dog out for a walk. A lot of these are habitual behaviors that are actually quite automated. And so in many ways, these standard behaviors and how to inhibit these standard behaviors can be trained on an habitual level. But of course, as you grow up and as you have more stimulation from the outside, then the habits are overcome by the lure of the stimuli around you. And, as you know, when little kids can succumb to that. So the marshmallow test is a perfect example, right? Everybody knows about the marshmallow test, right? You have the marshmallow there, and you have the kids, seeing the marshmallow, and then you tell the kid, okay, don't eat the marshmallow, get, 2 marshmallows later on. And it's this kind, and you can see that when you see these video images, there's all this kind of inhibitory function going on. A lot of it is habitual, and the cortex is really trying to kind of make sense out of that. It's very hard, obviously, as you know, to really study these 3-year-olds in in a brain imaging machine, because of course it's hard to put them in there and they kind of stay still. We're trying to do that now with 9 to 10-year-old, and believe me, there's some people in the back that will say, "Man, that's going to be a challenge." Eventually we're hoping to be able to have other techniques available that will be able to better tell us, how does the kid as young as that able to modulate that inhibitory system. And again, that is important to know, because then we can figure out a way of how to boost that under certain circumstances. We don't want to have a totally inhibited child. I mean, that's, the point is not to overgrow the inhibition. The point is to have adaptive inhibition, right? So sometimes it's fine to let go, and sometimes it's fine, it's important not to let go.
I totally understand, because I work with 3-year-olds and then I have a 14-year-old. So I'm like always in a neurodevelopmental habituation
Yeah.
myself. But I have a question regarding that. So you said boosting. So do you think the kids of children as young as 5 or 6 who are better if they are habituated or boosted with positive responses will do better as an adolescent to
Yeah.
get
So for the predictive test, the inhibitor system, the degree of inhibition early on really does predict adult behavior, at least within some of the impulsive actions like drug-taking behavior, things of that nature. So there's definitely that. Well, I want to again thank everybody for coming, and hopefully,
You are going to accept suggestions for future?
absolutely, yes.
Well, I am, I'm sure others as well. I'm very excited about the possibility of things that can enhance the B, D, and F and other
Yep.
things.
Yeah, I have a whole talk about optimizing the brain. So we can talk about that optimization of the brain. So that's a good suggestion. Well, again, thanks everybody for coming, and hope to see you in a month.